Okay, I'll get started then. Firstly, thank you for inviting me, Hugo, Natalia. I'm going to be talking about intracranial EEG. You have a scalp EEG cap here. Hopefully it'll be relevant. There'll be a couple of slides on scalp EEG and why this is a better option, although much more limited in access. But we'll get started. So an overview of what I'm going to be talking about today. I'll start with the most important thing, a bit about myself. Then what is intracranial EEG and how we use it in basic or discovery neuroscience and clinical research. And I'll end with neurotechnology and some industry-related applications. So how did I get here? Not today, just in general. This is my education pathway. So I started from psychology as well. Somewhere along the first year of psychology, I realized the brain was a bit more interesting. So I decided to go on to do a master's and then a PhD in cognitive neuroscience. I then took on a position in clinical neuroscience as a postdoc. That was my first introduction to intracranial EEG. I then got a Marie Curie, that's a bit cut, fellowship to work on aging. And around the same time, I started getting involved with a startup called Find Neuro. I'll tell you a bit more about it later on, where I was a computational data scientist, neuroscientist, and progressed into a more senior position. And just under 2 years ago, I started as a lecturer here and was awarded the Royal Society University Research Fellowship, URF for short, which basically means I'm a research-only lecturer. So what my research and my lab's research interests are, very broadly, how are memories formed? What are the cognitive and neural mechanisms that support encoding of information into lasting and durable memory? So this is something that you'll hopefully remember for a long, long time. Part of that has to do with the state of the brain. So you might have heard the term brain state if you've been looking at EEG. Very broadly defined here is how do we get the brain to attend to the external world, to process sensory information in order to improve the chances of whatever it is that we're experiencing becoming a strong memory. This is as opposed to kind of an internal state where you might be trying to remember what you had for lunch yesterday, but you're not really focusing on what's going on in the outside world. Secondly, as I've mentioned before, I'm interested in aging and what happens to our memory system as we age. You're all aware that as we age, memory tends to decline. But that's not the case for everyone. There's a unique group of people known as superagers, and they seem to be able to resist this age-related decline. So this is part of the work that I did in my Marie Curie fellowship, and I continue to do now with a PhD student in the lab, is to try and figure out what neural factors basically help these people retain their memory performance. So we're talking about people who are in their 80s, but perform like they're 50 or 60 years old. So that's quite remarkable. And finally, and more recently, I've become interested in how we measure memory in clinical populations. So that can involve the aging side of things if we go into the dementias or Alzheimer's disease. But more recently to do with epilepsy, as you'll find out in a second. Cognitive assessment of memory is a bit stuck in the past, partly because we need to be able to test memory kind of quickly and with populations that might not be able to do a full experiment like we tend to do in the lab. So for that, there are standardized neuropsychological tests. We give you a few words and you have to try and remember them. What we're trying to achieve here is to move from that to more targeted granular measures that would still be fit for the population, so a clinical setting, but build on the experience that we have from the lab. All of these things, if you've studied neuroscience, I hope this was a clue, involve the hippocampus that we have here shown in blue. Has anyone heard of the hippocampus before? Everyone. Okay, excellent. So that is kind of the common factor in my research. And as such, we want to be able to look at it, examine how it works. So as you can see here, it is a small shape, seahorse-shaped region nested deep inside the medial temporal lobe. It's part of the limbic system. And as you may already know, it is one of the most studied brain regions, basically of them all. So I'm not unique in my interest here. Some lucky researchers even got a Nobel Prize for studying it. That's the GPS cells, if you've heard of that. Yeah, so John O'Keefe and the Mosers, Edward and Mary Britt Moser, won a Nobel Prize in 2014 for discovering these GPS cells in rodents, but of course they've been found in humans as well. Yes?
Does GPS, like as in like actual sat-nav GPS? Or like
No, so this is they're called play cells. The official term is play cells. GPS is what the general public would know that as. So it's basically your ability to, or the hippocampus fires when in specific locations in the environment. And that's how you're able to track your navigation or your location. Yes, please feel free to interrupt me throughout if there are any questions. So in humans, we mostly study the hippocampus using functional MRI. You're all familiar with that method? Okay, excellent. There are some issues to do with signal detection from there because of the location and blood vessels around it, but we're relatively comfortable that we can detect both signal from there. It's nested too deep inside the medial temporal lobe to reliably get scalp EEG data from it. Be very wary of people who tell you that they can localize things with scalp EEG to the hippocampus. Although recent advances in MEG do allow that, there is a very nice, relatively new method called optically pumped MEG, OPMs, where you can put a little sensor inside the mouth and that will make it closer to the hippocampus, and that's how you record the signal. And finally, the hippocampus is also a hot zone for epileptic seizures. So let's dive into epilepsy. Everyone's heard of the disease? Yes. So it's a neurological disorder that causes seizures. Basically, what happens is there's some recurrent abnormal activity in the brain that can be localized to a specific region. So it can start from a specific region and stay there or kind of spread.
So it's kind of like a short circuit where the neurons are causing themselves to fire faster and faster and faster until they crash out.
Yeah, not necessarily faster, but more coordinated, yeah. Mm-hmm. Yeah. So that's the focal or localized epilepsy. There's also a generalized epilepsy where the starting point or the source is kind of either in both hemispheres or it's too difficult to say how many sources there are. So yeah, you can see this would be the scalp EEG signal. When it's focal, you can see that specific region will start firing, whereas the other brain regions will kind of look normal. In generalized, it's all over the place. So how do we diagnose epilepsy? In the early stages, we use scalp EEG or MEG to try and find or pinpoint the seizure onset location. This is usually combined with video monitoring. So detecting a seizure just from the signal can be a tedious task, as you will see in a second. Video EEG helps the clinicians align the time series from the brain with the timestamp from the video, so they can see that the patient's having a seizure, and then they can go in time on the EEG dataset and look where it started. The caveat of this early stage diagnosis is that, of course, if the source is deep, we're not going to be able to pick up the signals with a scalp EEG.
You have to wait for the patient to actually have a seizure as well.
Yes.
And so there's a chance you come in to get your recording done and you don't have a seizure.
Yes.
And it's kind of deep.
Yes. So monitoring can be up to a week with video EEG. Usually that's not the case. There are ways to induce seizures as well.
Are you allowed to do that?
In a clinical setting, I mean, not me personally, but yes. I'm not allowed to do that.
Like flashing light methods.
Yeah, some epilepsies are photosensitive. Others, yeah, there are other ways to induce it, but yeah. Yeah.
So what kind of seizures are usually caused if it originates in the hippocampus? So like, is it grand mal? Is it sort of tonic-clonic? What?
That's a good question. I don't actually know what would be the definition of that, just because I generally don't tend to. So what the data that we look at is if there's hippocampal sclerosis, so basically you have a structural lesion caused by the epilepsy. We tend to throw the data away because it's useless for research purposes. Yeah.
So during the EEG, you are trying to induce a seizure, is that?
For the clinical setting, you mean? Yeah, well, yes. So you're trying to record the seizure, let's say, first. And then.
What happens after if they do have a seizure, what happens after? What do you do?
You mean in terms of clinical plan? So they try and localize. So the reason why you're doing EEG is to try and localize the seizure. If you're able to, then the first phase would be drugs, would be medicine. If that doesn't work, you'd then get into intracranial EEG. But the plan here is to identify what kind of seizure that is, and then administer the relevant medication. So that's the case for 70% of patients. Scalp EEG is usually enough, and drugs seem to be able to allow them to live a relatively normal life. For 30% of patients, that's not the case. They're known as drug-resistant epilepsy patients or DRE patients. And in this case, the treatment most often is surgical. So you can see here a surgeon trying to put the robot to start aligning the electrodes in. There'll be a video in a second. So to help us identify the brain area to be treated in the case that it is not picked up by scalp EEG, the clinical team needs to basically put some electrodes inside the brain. Now that can be on top of the cortex. So we do a craniotomy, we open the scalp. We put a grid of contact or electrode on top of the cortex, measure activity, and then later on remove that, put the skull back on. A nicer approach for the patients is depth electrodes, in which case, and that's what you can see here and here, you make very small holes, burr holes are called, in the skull. You put an electrode through, and each electrode has multiple contacts that you can record the data from. This is now standard practice. In Europe, this has always been, I think, the more common option. In the US, this, for some reason, was more common up until recently when they've agreed that we call this stereo EEG because a depth electrode is one electrode. When you have multiple ones, it's like a stereo vision. But so this is now, the US is catching up with us. So let me show you a video of what actually happens in a surgical point of view. So this is from, this is pediatric. Do I need to stop sharing and then start again? Probably, okay. It's from a pediatric patient or process, so to speak, but adults go through the exact same thing. Display. Okay. Oh, sound would be useful.
To find out where the seizures are happening in the brain, a pediatric neurosurgeon may perform a procedure called stereoelectroencephalography, stereo EEG, or SEEG. A robot will be used to insert electrodes into the brain to help find where the seizures are happening. To begin, the surgeon will secure the patient to make sure everything is aligned with the robot. Then the patient is connected to the robot. Next, the robot will move to the precise spot to place the first electrode. Using the robot arm as a guide, the surgeon will make a tiny hole in the skull. Then an anchor will be placed in the hole. This process will be repeated until all the holes are made. Next, the surgeon will carefully pass an electrode through each hole into a precise spot in the brain. Each electrode will be tested for quality of recording brain waves. To end the procedure, the surgeon will remove the robot and securing device from the patient, but the electrodes will remain in place. The head will be wrapped with a bandage. After the procedure, the child will stay in the hospital connected to a video EEG. Over the next 3 to 10 days, the seizure activity in the brain will be monitored 24 hours a day. During this time, a pediatric epileptologist will work with the neurosurgeon to find the location of the seizure activity in the brain using signals from the electrodes. Once the recording is completed, the electrodes will be removed from the brain. This procedure takes 15 to 30 minutes in the operating room under general anesthesia. Based on the findings, a plan of action will be developed. This plan may include surgery to remove the affected part of the brain, stereotactic laser therapy, depending on the location and size of the affected area, or non-surgical treatment options.
Okay, so we'll cover some of these in a bit. Let me move back to... Okay. Questions about this procedure? Yeah.
Is it more standard to do these on children then?
Not more standard, but as standard. So
Statistically more.
Yeah.
for children.
No, not necessarily. It's just you can do it in children as well.
Yeah.
So obviously, if you realize that drugs are not working, you might as well do it as early as possible.
Do you think in the future there'll be less invasive ways of doing it? Just as like an observation, like that looks terrifying.
So yes, in reality, when you see the patient accept the fact that they have a big bandage and something coming out, it's fine. So they wear a bag, like a side bag, that carries the amplifier. They can't shower, but they can go to the toilet. Obviously, they have their own room for the week or for however long we have them. Let's get back to that when we get to Neurotech.
Okay, cool.
We're making some steps, but it's probably not.
And another slight thing, like when you implant the electrodes, does it damage the neurons as you're passing it through?
No, it's really, really carefully done. So let me walk you through. So you're not gonna ideally damage anything. It's just like you're putting something through jelly, so to speak. So this is a reconstructed image. So that's an MRI image. I put a nice volumetric hippocampus here. And it's a reconstructed electrode with the contacts in as circles. So that's a depth electrode. Each electrode will have multiple contacts. So that's an actual electrode that we put in the brain. Each of these silver bits are contacts. You'll get anywhere from 6 to 12 or 18 sometimes, depending on the contact itself. And what we're sampling here is local field potential. So this is some synaptic activity, roughly 2 millimeters in radius. 2 millimeters is usually 2 to 3 millimeters is the distance between each contact. Questions?
Yeah, I think my next question is going to be how many electrodes do you tend to use?
Yeah, so again, purely clinical need, anywhere between 8 and 12.
Okay. And are the areas which these electrodes are going to cover like pre-decided?
Yes. Yeah, so the epileptologist, who is a neurologist, will consult with the neurophysiologist, who is also a neurologist, and the brain surgeon. They'll come up with a surgical plan. The surgeon will then put the electrodes. You'll take a CT image to see where the electrodes are. You can overlay it on the MRI to end up with something like this, to confirm that they're in the right spot. We kind of assume all of this is linear. The electrode does happen to sometimes go in a bit wobbly. That's fine, there's not much we can do with that, but usually you're able to pinpoint at least I'm in the anterior hippocampus, I'm in the posterior hippocampus, I'm in fusiform gyrus, etc. So in some centers, although again, less common in the UK because this is purely for research purposes, it serves no clinical meaning, the electrodes will have microwires. These are the tiny kind of hairs at the end of the electrode that is able to pick up action potentials from a single neuron or multiple unit activity from a number of neurons. That's much more common in continental Europe and in the US. In the UK, I think, if I'm not mistaken, it's still not been approved for use, also, although there have been conversations about making this available in the near future. It basically allows us to bridge cellular neuroscience, so a cell is firing. With a systems level, this is what the brain is doing, and cognition, as you will see in a minute.
So is this kind of like the human analog of the single cell recording that you do in rats.
Yeah, exactly. So we talked about signal sources earlier, deep in light blue and superficial in darker blue. Scalp EEG doing okay-ish, not to discourage you from using that.
We
but
know it's terrible.
iEEG is the grid that sits on top of the cortex. Excellent with superficial, not so much with deep. And then electrode, depth electrode, excellent with deep. You're not going to get any superficial. You can have concurrent scalp EEG and intracranial EEG if there's a need for a clinical need. Right, so how do we use intracranial EEG in discovery science? First, a caveat. We're limited by coverage that is to say clinicians are the only ones to decide where the electrodes go. We unfortunately can't come and say, oh, I want to see what the hippocampus is doing in this patient. Please put an electrode in. So with that caveat, still intracranial EEG provides the highest spatial and temporal resolution in human neuroimaging. So we're talking millimeters in space and milliseconds in time. So that will depend on the sampling frequency, but usually it's 1,000 hertz, so 1 millisecond per time point. As Abia said, it allows us to directly compare or translate findings from animal models, so this can be rodents or non-human primates, into humans, which is incredible. And depending on the regions of interest and where the electrodes are placed, we can look at connectivity or brain networks as well. So we can see contact number 1 here is in the fusiform, and contact number 10 here is in the occipital cortex. What happens during some cognitive function? How do they correspond, or how do they talk to each other? So because I've preempted everything with the hippocampus, I will stay in the hippocampus. I just wanted to show you an example analysis. So this is from my own work, which I'll get back to. But what you can do with the data, essentially, so on the x-axis across the board here, we have time in milliseconds. Sampling frequency here is 500 hertz. So each time point is 2 milliseconds. We can do a time frequency analysis. Do you guys are familiar with time frequency? Yeah. Basically, it's a fast Fourier transform. So you get the signal into the frequency domain, and then you can see which frequency is the most prominent. That would be power. This is an example of a raw trace from the hippocampus and filtered to what we call ripples between 80 and 120. So you can see this nice squiggle. Okay, so an example, we'll start with work that's not my own. It's Alex Vass' 2020 Science Advances paper, where they looked at single neuron activity. And here they're interested in replay. So this is if I show you a list of words, for example, and I ask you to remember them, when you're retrieving them, is your brain actually just replaying them, so to speak? And that is essentially what they show here at the single neuron level. So whatever happens at encoding ends up following backwards at retrieval. If your neurons are firing following this replay sequence, you're more likely to be correctly remembering the next word. And this is from my own work, and the point here is just to show you the communication between different structures. So we were interested in the hippocampus, and again, these ripples that are basically a marker of highly synchronized activity that the hippocampus uses usually to replay things. So this has been highly or very strong evidence in rodents. In humans, it's becoming to be more and more apparent with more of these studies using intracranial EEG. What we've shown here is that it's not just for replaying what has happened, it's also used to pre-play what we think is going to happen, so in planning. So what we show is that whenever the hippocampus does this thing, usually it's in uncertain context, so when we don't know what's going to happen. And if we see this thing happening in the hippocampus here, it then triggers some response in earlier visual cortex. So in the fusiform and in the occipital cortex. So we've shown this direct communication between the two. This is kind of feed forward. And there's also feedback. So when something surprising happens, we've shown that the fusiform tells the hippocampus, work a bit harder now, something unexpected happens. So the point here is to show you that although we have limited coverage, we can treat the brain at the region of interest kind of level. So we have multiple electrodes here. It's not that we cover the whole cortex. Okay, questions about discovery science. I realize these are just 2 examples. There are plenty more. Yes.
I just had a question about what's the measure of entropy? Because in the previous...
Yes, it's Shannon's entropy, so it's information theory entropy.
Okay. Could you explain a little bit more what that means?
Yeah.
Hopefully it's not too...
No. Well, if it's of interest, yes. So entropy quantifies how uncertain we are about something that's going to happen. So in this case, we had a series of stimuli. There are 4 colored shapes. And what we're quantifying is how likely, so to speak, the next shape is to be based on what happened previously. So if you've seen green triangle, green triangle, green triangle, green triangle, probably the next one is green triangle. Entropy goes down. So the more experience of the same information you have, entropy will go down. If it's highly uncertain, so you see green triangle, purple square, purple triangle, green square, kind of all over the place, entropy is high, it's quite uncertain. So it's basically trying to find this structure in the environment. And when we don't have this structure, uncertainty goes high. That is what entropy is.
So if you have high entropy in the environment and high different possibilities of what you're going to experience, you see, again, higher entropy in the brain activity?
We relate entropy to the brain activity. So we're not measuring entropy in the brain. We're relating entropy in the environment to brain activity.
Because in one of our modules, they started talking about, it was the psychedelics module of neuropharmacology.
Okay.
And they were talking about like brain entropy states.
Yeah.
And like default mode network. But like just using very generalized words and not really explaining what any of it meant.
Yeah, so if you take, can you remind me when we get back to the virtual epilepsy patients? And I can show you how that can be done. Sorry. Any more questions about discovery science? No. Okay, great. Clinical practice. So obviously, intracranial EEG, you need that for localization of seizure onset zone. That's the brain area that is the culprit in generating seizures. Some clinical terminology for fun. They don't call this epileptic activity just one thing. So you have ictal, which is when the seizure is happening. You have pre-ictal, preceding the seizure, post-ictal, following the seizure. That will become important when we try and predict seizures. Yes?
Can I ask, what's the link between the idea of entropy and the seizures?
Oh, no, that's unrelated.
Oh, okay. Okay, and because you mentioned that you can link entropy to the brain activity.
Yeah.
So can you see, like on the graph?
Well, not on the graph, but what you will end up seeing is more of these.
Okay.
So there's more of these markers in high entropy contexts. Okay, so we talked about recording from the electrodes, but they're not just there to record, they can also stimulate. And that is useful in the clinic for a number of functions. The first one is to map cortical connectivity or networks. So this is done using low frequency stimulation, so it's 1 hertz usually. What happens, let's say you think this is the language network for this patient, you can stimulate a specific contact, number 7 here, and look at how the other contacts respond to that stimulation. So the gray shaded area here is when the stimulation is happening, and you can follow activity in time. Then that will allow you to create a map of how these regions are connected. So we usually want to spare language, memory, attention functions in these patients. So it's useful to know what the network looks like. The second type of stimulation is a high frequency one that's at 50 hertz, so a much faster one. And that allows us to induce a seizure. So we can stimulate the region that we think is the seizure onset zone and see whether that indeed generates a seizure. It can also be used to tell us what happens if we remove that region from the brain. So if that is the seizure onset zone, let's say the hippocampus, and we do need to treat it, and the treatment plan is resection, although that's less common nowadays, we can stimulate it, which will essentially inhibit its activity. and see how the patient performs cognitively. So we can give them a task, a memory task, and see whether they're able to remember or not. So in this now relatively old paper, but one of the co-authors is the neurophysiologist at Northern Caroline's at Salford Royal Hospital, they've shown that there's a material specificity effect with the left and the right hippocampus. So when they stimulate the left hippocampus, memory for words went down, but everything else kind of stayed the same. Whereas when they stimulated the right hippocampus, memory for faces went down. So it can be used for research as well, but of course this is more clinical than anything else. Some points to consider about intracranial EEG. We talked about sampling bias. We don't decide what the electrodes are. Generalizability. It's a small number usually of patients in a study, anywhere between 10 and 20, unless you're in the US and it's a massive money-making machine and you have hundreds of patients. And epilepsy, of course, is an inherent confound. If we want to study cognition, are we sure that the epileptic brain is the same as the healthy brain? For clinical research, epilepsy is a network disorder. So there's starting to be a move from looking for those specific seizure onset zone to looking at the network of regions in the brain. Again, that is masked by the sampling bias. We only see what we look at. We cannot put electrodes in the entire brain. We might be missing a critical node in the network if we haven't placed an electrode there. This is where virtual twins will come into play in a few slides. And seizure prediction, we talked about that pre-ictal period. Some people are interested in trying to predict, based on that, will a seizure happen or when is that pre-ictal period? Can we stop it? That will become useful in a few slides as well. So neurotechnology, finally. It's a hot topic. This Nature Reviews Neurology 2024 paper, they call it AI in epilepsy. You'll excuse me if I don't call it AI. It's more machine learning models than anything, but it is a grand challenge, and a lot of people are working on it. I've listed some questions, but there are plenty. One of them is, can we detect seizures in real time? Can we predict seizures? Then it's the next one. Is the seizure onset zone really the most critical aspect in the network? Or if we remove some other node in the network, would that be more useful in stopping propagation of seizures, for example? That becomes important if we can't operate on that seizure onset zone. So if this is an area that is very important for language, we don't want to leave the patient without any language abilities. We need to be flexible in our treatment plan. Yes?
When you're doing seizure detection, is it just, are you detecting whether or not a seizure is going to actually exist and happen, or is it the latency after the pre-ictal period that it'll take for that seizure to occur?
No, whether the seizure happens. Okay. I mean, you can try and predict the latency and so on, but you first need to be able to say whether it's going to happen or not before you start characterizing it. Okay, there's a lot of text, I apologize. I will just talk about the figure and you can read the text if it's useful. So we talked about sampling bias. We're not going to put electrodes in the entire brain. There's a new cool thing. It was reviewed in the 2023 Lancet Neurology paper called the virtual epileptic patient. Has anyone heard of digital twins? Kind of, okay. So it's an approach whereby you create a model of disease. Let's just stick to the brain, a brain model that you can play around, that you have sufficient data to have the same level of accuracy as a real brain, but you can play around simulating data and see what happens. So to create this virtual epileptic patient, we take a real patient's brain imaging data. So this will be structural and functional MRI to basically allow us to reconstruct structural connectivity in the brain. That's white matter tracts, how they're connected. Functional connectivity, how these regions talk to each other. In some cases, we also have their intracranial EEG, depending on availability, but that doesn't really matter. The point is that when we have these type of information, we can start creating these nodes in the brain that represent brain regions. So we parcellate the brain into different regions. The amount varies. Each region becomes a node in the network, and how the nodes are connected is defined by the structural connectivity and the functional connectivity of the patient. Then we end up with this mess. Each node is basically a computational model that represents the average neuronal activity. This is known as neural mass models. we can now say, okay, let's say I think that the seizure is coming from here. Let me simulate a seizure and see how the rest of the network behaves. We generate some predictions. We might come up with what we think the seizure onset zone is. We can test it in the patient's brain to see whether we're right or wrong. Yes?
So what is generating the predictions?
Well, the simulated data. So you have some hypotheses usually based on where the electrodes are. But this allows you to generate seizures outside of the patient's brain.
So that does sound a bit machine learning.
It is. Well, it's computational modeling because how the neurons interact is a biophysical model usually. But yes, generally speaking, it's not AI. It's not a black box.
So here they've compiled a whole bunch of information about different brains from when they've done imaging on them and so on, and created one model that fits everyone nicely, or you can tailor
So that's
it
one
a bit?
approach. Or if you want a digital twin, if we follow the definition carefully, it's that patient's brain. So it's a one-to-one.
And then you can keep messing around with that patient's digital brain.
Exactly.
To see how it goes.
Exactly.
That's pretty cool.
What is the general accuracy of that? Like how good is the predictability?
Excellent question. I don't know.
No worries.
To the best of my knowledge, well, I don't know how much they've tested this on real data. So it's a bit circular at this point because we do have the real data, right? And if we do include that in our virtual brain, it will change from patient to patient. So the added benefit of it needs to be kind of in a prospective study, right? So that's very dangerous. Imagine I build this virtual brain model. I say that needs to be removed, but without any intracranial EEG, we just go and remove it. Dangerous, but it's going there. Yes.
Well, like you can only get limited data from the real patient. So how can you make sure the model always goes right? So it's more like a black box.
So we're now at the stage where we still use intracranial EEG to verify the model's predictions, right? Decisions are based on this, not on simulated data. Does that answer the question? There are a few hands. Okay, yeah.
So do you use the simulated data to generate a hypothesis, or do you test a hypothesis with the simulated data?
That's a very good question. I want to say that you generate an hypothesis, but given that you already have the data, it's a difficult statement to make.
This has more to do with the last slide, I think. For the seizure initiation, could we potentially use neuropixels? Because we kind of talked about it in one of our modules last semester, to predict seizure onset, maybe. I know that there's no human models.
Yeah,
Nice, but just
I don't know if the rodent literature does that.
yeah.
There are epilepsy researchers in the rodent world at Manchester that they might know better than I do. We don't have neuropixels for humans, so
Yeah. Just the
maybe. Yeah. Any more questions? Virtual epilepsy brain? Yes.
I had inverse. It was further back to do with the site selection bias type stuff. So are you usually working with a single site to get your patients that are going to give you data, or are you doing it kind of across multiple sites? Or
For research, it depends how lucky you are. If you have good collaborators, you can have multi-site.
I was kind of curious because if you're working across multiple sites, how different is? Because I would have thought there might be relatively standard, but there are also different practices might have. They might prefer, we prefer to put our electrodes in these positions.
Yeah.
I was curious how that kind of affects your
Yeah.
comparisons for.
So that's why we tend to work with regions of interest. So if I go a few slides back, sorry, more than a few slides, back to the hippocampus here. So these dots represent contacts across patients, across 2 sites in this case. We've selected only anterior hippocampus, which is why you don't see anything in the posterior. But we treat all of them as if they originate from the hippocampus, not from a specific location within the hippocampus to solve that problem. But you're absolutely right. Yeah, sampling in terms of space within the brain as well is going to be different. Any more questions while I jump through? Right, okay. Okay. This is going to solve a lot of the issues that we discussed, hopefully. It's a very promising approach in treatment. No, don't navigate.
Neuromodulation for epilepsy.
Display. I will warn you, this is from an American center that obviously makes money off this treatment. So although it's accurate, it might seem a bit promotional.
Neuromodulation for epilepsy involves implanting devices either into the brain or on the vagus nerve of the neck. Unlike traditional surgeries for epilepsy, which involve removing brain tissue that causes seizures, neuromodulation allows us to implant devices in the brain that stimulate and reduce the likelihood of seizures starting and spreading. In many cases, these types of surgeries can have less of an impact on functions such as memory, attention, or language, for example. Neuromodulation allows patients to reduce their seizures and in rare cases can have the effect of making patients seizure-free. The three types of neuromodulation for epilepsy are vagus nerve stimulation, or VNS, responsive nerve
Much less common now.
RNS, and deep brain stimulation, or DBS.
More for Parkinson's
Responsive
than
nerve
epilepsy.
stimulator, or RNS, has two main parts. One of them is a generator, which is implanted in the skull. This sits flush with the skull and is back behind the hairline. The other pieces are electrodes. These are placed either into the brain tissue or they sit on the surface of the brain.
Depth
The
or
RNS
grid?
electrodes constantly monitor activity in the brain. When they detect the signals that indicate the onset of a seizure, this triggers the device to deliver stimulation. That responsive stimulation is designed to stop seizures just as they are beginning.
So the pros for the RNS is we can long-term monitor patients. So for example, when patients have more than one area of the brain that the seizure is coming from, we can find out what percentage of the seizures are coming from site A and what percentage is coming from site B. Then oftentimes we'll go back and do surgery for that patient. Neuromodulation offers hope for patients in whom resective surgery often is not an option. Our team of neurosurgeons and epileptologists work with our patients to determine what their goal is from epilepsy surgery, from coming to us, and we help them navigate through this process, no matter how long it takes.
Yeah, no matter how long it takes. So that's exciting, right? Forget about the, the promotional side of things. We don't have to remove any area of the brain. We can predict when a seizure is going to come and stop it. That is really cool because the doctor can track the data with a Bluetooth machine from that thing. So you put like a...
You'll have an afterwards. So
Yep. It is used in research as well. So this makes naturalistic neuroscience a reality invasively. So the patients will walk around with this massive backpack. They will be recording from inside the brain, but they can move around in the world. They can come to the lab. They can walk outside. They can do whatever. So it gives us the opportunity to record local field potential or action potentials from behaving humans who are not necessarily in a hospital. Okay, I'll wrap up with what I've done in Neurotech. As my role in Find Neuro kind of evolved over the years, I got involved because the CEO is a good friend of mine, CEO and founder. He contacted me one day after I presented a poster at a Society for Neuroscience conference and said, "Oh, you're working with epilepsy stuff. You want to come and do some real-world impact work?" I said, "Sure, why not?" So the first thing we looked at is this electrode localization. So I've shown you the reconstruction of an electrode. Usually it involves some manual verification of where the electrode and the contacts, more importantly, are, because of course we only know where the electrode is from an artifact on a CT image that's resolution is quite low to begin with. So we try to build an algorithm that is able to do that as accurately as a human neuroradiologist would be. We've also looked at using existing data to create a database that talks about different sites. So in the US, this is an American neurotech startup. In the US, some hospitals will only see a few patients like this. They might not be as experienced. We wanted to help them benefit from more experience in larger sites like Mass General Hospital, which is where the startup was a spin-off of Harvard MGH. So we built, or we were working on building this database that will allow clinicians to find similar patients. So this patient had a similar clinical profile. This was a surgical procedure that they took, and surgical outcome 12 months after. So if surgical outcome was positive, the patient had a similar clinical profile, maybe you want to take the same approach. So there's some modeling of similarity there. And finally, treating epilepsy as a network. This is, we've been working on trying to use graph network and information theory measures to predict where is the most critical node in the epileptogenic network. And I wanted to end with some hope. So in the startup, this is right before I started, I think, actually, so I wasn't involved in this, but there was a patient who had a lot of seizures. The predicted surgical treatment for her was different from what our algorithm suggested. We managed to reason enough, or they managed to reason enough, I guess, with the surgical team, and eventually, if they removed the part that our algorithm suggested would be the critical point, and her seizures stopped completely. So there is real impact here. So with that, I'd like to thank my collaborators at Noam at Fine Neuro and clinical collaborators at Northern Care, funders, and you for your attention. Any more questions?
I have a question.
Yes.
So, you mentioned that it's possible for different new approaches to find the central node that then triggers a seizure. I wonder if it would also be possible for, let's say, two nodes to be critical, whereby one is conditional on the other and the other is conditional on the first one, and together they create this loop that generates the seizure.
Yeah, so that's exactly the move towards a network. Because if you're trying to locate where is the first contact that the seizure activity started from, you might be missing that, right? So you remove that, okay, but then that next contact might be the more critical because it helps propagate through the network. So yeah, that's definitely a possibility.
So would it also be possible to find the kind of least useful networks, or the least useful node in the network that nevertheless creates the most seizures, and then remove that instead of maybe the optimal one, but that removes the function that you might need in the brain?
In theory, yes. In practice, I don't know whether that is a real scenario. I can't imagine what would that look like. But yeah, so if you start using graph network analysis on these, you can look at inputs to a node and outputs from a node, right? And then it becomes you have a lot more information that this is where it started. Yeah.
I had a question because you mentioned that it was a colleague of yours that started
Yep.
Neuro. Do you know his career trajectory? Is he coming from a neuroscience background, or was this?
Yep.
Yeah.
Yeah. Well, undergrad he did in maths and biology. But yeah, neuroscience, master's, PhD, postdoc. So he was a postdoc at Harvard when he started this.
Is it more typical for maybe people from neuroscience go on to start these startups and then reach out to researchers to check the quality of what they might be producing, or join a team, or is it more the researcher's job to seek those opportunities? Do you know?
You mean in terms of how do I get a job at a startup? Okay. Yeah, so, I was lucky enough that he knows me and we've been working together for a long time. So for me, it was the other way around. But yeah, it is more common. If you are interested in joining work like that, it's more common for you to go and approach them and say, I want to work in this field. Are you hiring, or are you considering taking on interns?